Drug discovery. Drug discovery is basically an activity to find out drugs which are used for prevention and cure of diseases. Now this drug discovery is a time consuming task and have several parameters. Parameters means that it has different parts, such as we need what would be the target drugs or what would be the lead components which will be binded with the drugs to attain some cure or disease prevention. Similarly, what we have seen is that bioinformatics can be a very good resource for generation of different drugs. How it is a good resource? Because in bioinformatics we have been focusing on genes, and then there is a lot of information available to separate genes, and that this information can be a milestone or useful thing for generation of different drugs. So let us start from a bit of history related to drug discovery framework. Before 1960s, drugs were usually a molecule which was applied on the complete human body, and there was no information about particular genes which were present to remove that disease from the body. So body was considered as a black box, and then a medicine was given, and then there was a change in the complete body of the organism. In 1970s, we saw the emerging of mechanistic diseases or devices, which was basically different receptors and enzymes which helped us to identify different parts in the molecules. Later on in 1980s, we got separation mechanisms such as liquid chromatography, GLC basically gas liquid chromatography, and mass spectrometers. So what happened is that scientists were able to separate different ingredients of molecules. Once they were able to identify different ingredients, they were able to identify which genes were responsible for curement or causes of diseases. And after 1980s came the era of high throughput computing. This high throughput computing was done in medical or drug discovery domain by HTS, basically, which is high throughput screening. So high throughput screening, basically, instead of working only on one gene at one time, we can work on millions and billions of genes at the same time and in some laboratory parameters. Human Genome Project. Human Genome Project was basically a USA based project which completed in 2001. So purpose of this project was to annotate all the genomes in a human organism. So what was the effect of this project was that it had very good and long lasting effects on the drug discovery field. Since we came to know most of the genes or genomes inside the human body, so once we know all the genes or most of the genes, then we can use these genes for the discovery of different drugs to attain or remedy different diseases. So basically, goal of this project was to complete or to generate complete mapping and understanding of all the genes inside a human gene. Once this information is attained, it is used for drug discovery type things. Now, what is drug discovery? Drug discovery is basically a decision making process which is based on huge amount of scientific data, and that data is used for identification and validation of associated genes for specific disease. So what are different challenges in drug discovery now? The most important challenge is compound selection. Compound selection means that which compound would be useful for generation of a specific drug. Then we have high throughput screening challenges, that we should have mechanisms so that we can map or we can check millions or billions of genes in one go. Then we should have a mechanism for ADME and toxic information. So ADME is basically absorption, distribution, metabolism and excretion of information from a body after drug intake, and then they shouldn't have there any side effects, such as there should be no toxic effects or there कोई जहरीला इफेक्ट नहीं होना चाहिए मेडिसिन का. Then drugs can be monogenic or phylogenec, so polygenic basically. Monogenic are those drugs which are based on single gene or single gene diseases. So, or second one is polygenic. Polygenic are those diseases which are comprising of multiple genes. When we say polygenic diseases, there is also a cause and effect relationship. Cause and effect means that one gene will affect the other gene, and then we have to understand and working of both the genes or more than two genes to reach some assumption. Then we have in vitro and in vivo data testing. So in vivo means inside the living body, and in vitro means inside some glass tube, or we can say that in vivo means that we are checking our medicine on direct target, and on the other hand in vitro means that we are performing our experiments in sub lab. So these are different challenges which should be focused while we are working on drug discovery mechanism.